So hi guys, today we're going to talk about this smear. So what is this? So what do you understand by the thick and thin smears in malaria? So bacha log, whenever we talking about thick smear, what do? Is we keep one drop of blood and we do not fix it. Okay, so we do not fix it in methanol. We directly stain it with Giemsa stain at the pH of 7.2. So when you're doing this, what are you doing this? So you're basically lysing all the RBCs. So when your RBCs get lysed, obviously what you are seeing is just the malarial parasite. So you are able to identify the malarial parasite easily. Whereas, what do you do in a thin smear? In thin smear, you make a slide, you fix it in methanol, and then you put a Giemsa stain on it, and then you stain it at pH of 7.2. So the point is, now because you've fixed it, now the red cells will not get lysed. So you will be able to identify the parasites inside the RBCs. So you can identify the species and the morphology of malarial parasite well. So I hope everybody can answer this question now. So you know that in thick smear, the red cells are lysed. Okay. So that is the importance of thick and thin smear. Now what is the importance of pH? Remember, the pH that is preferred for malarial identification is 7.2, because at this pH, the differentiation of the parasite from the red cells is very easy. Okay, so that is why you are able to see the gametocyte, the trophozoites very nicely. Okay, so the pH here should always be, in malaria, the pH should always be 7.2. Now the third point that you have to remember is, how does the Vivex enters into the red cells? So remember, the Vivex enters into the red cells with the help of Duffy antigen. Whereas when we talk about falciparum, how does falciparum? Falciparum is very adaptable. So it enters into the red cells through two pathways. One is sialic acid pathway, in which it uses glycophorin. And the second pathway that is used is sialic acid independent pathway, which is actually your CD147. So this is also called as basigan. So this CD147 is a common target for development of malarial vaccines. Okay, so that is again an important question that can be asked. Okay, apart from that, the last is the point of care testing, guys. So now we have point of care testing for malaria. So it's like your just strip test for testing. And if you're suspecting falciparum, we predominantly, what is present in it is HRP2. Otherwise, for both the species, LDH or aldolase can also be used. So that is the important point. So that is the point of care testing of malaria. So these are the common questions that can be asked in malaria. And never forget the banana-like gametocytes of falciparum. And black water fever is seen in falciparum malaria. And what is the granuloma of falciparum brain called? Answer in the comments below. Bye.